Well, good afternoon, everyone. I'm Eric Green, Director of the National Human Genome Research Institute, and I want to welcome all of you to this annual lectureship, the Jeffrey M. Trent Lecture in Cancer Research. I'm going to give a general introduction to this lecture series, and then I'll turn over the podium to our Deputy Scientific Director, Paul Liu, who will introduce today's speaker. But I have the pleasure of saying a few remarks about this lectureship and the person that this lectureship is named after, and that's Jeff Trent. The story goes as follows. If you go back in time, the intramural program of NHGRI has not been around forever by any means. And in fact, 23 years ago, it really only existed as an idea. But Jeff Trent was brought here to essentially build the intramural program. He was brought here jointly when Francis Collins came here as then the director of the Institute, and was asked to create an intramural program. That is not an easy thing to do when it doesn't exist at all. He had to start from scratch, recruiting a number of people, getting space, getting staff, getting everything organized. And what I would say is he did it in a spectacular way. I had the good fortune of joining the effort not in 1993 when Jeff arrived and some of the first investigators arrived, some of which are actually in the audience. I arrived a year later, but was very fortunate to be able to join what was already a growing and incredibly exciting place to be an investigator doing research in. And a lot of the credit for that really goes to Jeff. He was able to not only recruit good people, but to put together an organization and a style for our intramural program that I think has served it extremely well as it has progressed through the years, now actually in its third director of the intramural program. So Jeff was the scientific director from about 1993 to 2002. At that time, he departed and became the founding president and research director of the Translational Genomics Research Institute, or TGen, in Phoenix, Arizona, and then had a similar opportunity to build something spectacular out there in the desert. And I'll leave it to others to talk, and you could read more about TGen, and there are some great things that have happened since then. What I would say is I feel very fortunate to have watched Jeff lead the intramural program, and I think I learned a lot, as did all of us who are research investigators in the intramural program during his stewardship of the program. And then I was fortunate enough to then be appointed to be his successor, and I served as the scientific director of NHGRI from 2002 until just about 6 years ago when I got this new job. But what I would say is one of the very first things I did back in 2003, shortly after becoming the scientific director, was to honor what I thought was a very valuable legacy that Jeff had left behind in terms of his mark on the intramural program and being the founding scientific director. And we established this lectureship in his name. And so and meanwhile, we've had no problem getting truly outstanding scientists to be honored by coming and giving the annual Jeff Trent Lecture. Just to give you a flavor, people like Janet Rowley, Lee Hartwell, Harold Varmus, Mike Stratton, Eric Lander, Brian Drucker, Carol Greider, Charles Sawyer, Chris Amos, Bert Vogelstein, Stephen Chanik. Among that list are 3 Nobel laureates and multiple members of the National Academy. Needless to say, I think people in the community also have deep admiration for Jeff and for what he did here and are absolutely willing to say yes when we invite them to give this annual lecture. And sometimes what's also great about this lecture is that it gives an excuse for Jeff to come back and visit us for a day and spend time with us. And indeed, this is one of the years he's been able to do it. So Jeff is here along with his wife, Dee, and a pleasure to have you here, and thanks so much. So that's the history of the lectureship. And I'd like to turn this over to our Deputy Scientific Director, Paul Liu, who's going to have the pleasure of introducing this year's Jeff Trent Lecture. Paul.
It is truly my great pleasure to introduce John Carpenter. First, I want to say Dan Kessler, who is the current scientific director, sent his regret that he couldn't be here. He's actually in Australia. You know, it's just 13 hours ahead of us, and so he couldn't give this introduction. John probably don't need introduction to many in this audience, since he was one of the first recruited by Jeff into his lab as a postdoc fellow and then a senior research fellow when John graduated from Ohio State University with his PhD. And he came here worked in the Jeff's lab for about 6 years, and then he became a tenure-track independent investigator in 2000. Then he left NIH, went to TGM, became the professor at the Division of Translational Genomics, and the Division of Integrated Cancer Genomics, and also became the Deputy Director of Basic Research in 2012. Only recently, he moved to University of South California, become the chair of Department of Translational Genomics, and also newly formed Institute of Translational Genomics. John has made many seminal contributions to cancer genetics and genomics, and he has published over 150 papers, and many of them highly cited. I just checked this morning, one of them was cited more than 1,000 times, and which is quite impressive. He has several patents, and he's a very busy person. He's on so many committees, and he served NIH, on study sections and other, advisory roles. And it's really great pleasure to welcome back. Thank you, Trent. Mm-hmm.
All right, thanks. Thanks, Paul. Eric, I didn't know about that list. That's kind of scary. Wow. Well, I think I can say honestly that, no matter how far my career goes, I don't think there could be a more honorable place that I'd like to be than right here, standing at the podium, giving this lecture today. I have so many friends and colleagues in the audience, and a number of them who've, been incredibly supportive and have played very critical roles in some of the scientific discoveries that we've made through the years. And I won't call names because I don't want to miss any names, but to all of you, I just want to say thanks again. It's been a fun ride. I remember one of the Harry Potter movies, they were driving through London in a double-decker bus, and it was going all over the place, and the driver said in a very Caribbean way, "It's going to be a bumpy ride." And I wish people had told me that before I started, because it has been a bumpy ride, but I wouldn't trade it for anything. I have had the opportunity again to work with a lot of amazing scientists, and just want to say before I get started that I'm presenting this on behalf of a lot of great people. I don't take singular credit for a single thing that I've done. It's just not my approach to getting things done. I was sort of raised in the culture of team science by folks like Jeff Trent and Francis Collins. By bringing together, enough smart brains, you can get a lot of incredible things done. And so through my career, I've done a lot of work both on the germline genetics side, the cell biology side, as well as cancer genomics and tumor profiling. And I could talk about a lot of different things, but I really want to, bring to bear some of the work that we've done and some of the concepts that we're seeing moving forward, and how aspects of population and tumor heterogeneity have significant impacts on cancer genome science and clinical phenotypes. And I'm going to spend a lot of time again in cancer and in the new medical paradigm of precision medicine. Here again, focusing mainly on the work we've done in the oncology space. And I affiliate both with the University of Southern California as chair of the new Department of Translational Genomics, but also maintain a relatively strong relationship with Jeff and the team at TGen as an adjunct faculty member there. I know it's not AACR, but I always like to do this, and there are some financial disclosures. I'm one of the founders of Ashine Analytics, which is a clinical sequencing laboratory, and I'll discuss off-label use of several FDA-approved drugs, including ponatinib, pazopanib, erlotinib, and pertuzumab. So getting right to business, I think my presentation will be sort of sectioned based on looking at historical studies and some of the work that we've done, and looking at population heterogeneity as a function of cancer genome science, and then I'll go into some aspects of tumor heterogeneity and how that can influence our interpretation of data that's being used both in the research space as well as in the clinical space through precision medicine. So, in thinking about population heterogeneity, I think we want to focus on phenotypic differences that we see and looking at sort of global cancer statistics for incidence and death rates in men and women, and seeing the common cast of characters for incidence being prostate and breast and lung and colon and some other diseases that we don't look at very commonly because they're a little bit more rare, like bile duct cancer, cholangiocarcinoma, and then looking at death rates, again, lung cancer still being a very significant contributor to cancer deaths, even with the advancements in smoking cessation and prostate and breast, of course, and colon. And then we start to see tumor types that actually come off as being kind of rare, like pancreatic, and again, this bile duct cancer. I'm going to talk a little bit about the work we're doing in that space. But then when we focus on and look at cancer incidence and death rates in African Americans or blacks, we see a similar picture where we see prostate and breast and colon, but then we see things like bile duct cancer actually float up a little higher to the top of the list, and looking at looking at men, African American men, and it actually happens to be the fourth most common cancer in that group. It's not that far down the female list either, but again, just thinking about there are some commonalities when we look broadly at cancer statistics, but there are some differences in these things that we tend to look at as cancer health disparities, where diseases like prostate cancer are about twice as commonly diagnosed in African American men. And although breast cancer is more common in white women, the cancers can be detected a bit earlier and have more aggressive biologies in African American women. And I'll start off, talking about, these concepts of race and ancestry. And I see Vince Bonham, at least he was sitting in the audience there. And Vince has really spearheaded a ton of work and really trying to educate the population on these differences, and that these two things are not necessarily the same. And there's a really complex interplay between the two of these things, where race is being a social construct, and I think Francis has called it a proxy of sorts, and is much more related to social and societal related factors, where we have, and then we have ancestry, which is real genetics and biology and is associated with ancestral genetic material that's passed through individuals. And again, there's an interplay here where undoubtedly, individuals of, that have common ancestral backgrounds can definitely have societal clustering and higher interactions and can have more ethnic similarities. But then you can also look at ancestry. We can have individuals who have very similar genetic ancestries, but have very different cultural lifestyles. And so these two things, there's a complex interplay there, but I really want to focus on how we look at these things, and primarily, again, thinking about genetic ancestry and principal components analyses, and individuals like Rick Kittles and others have really spearheaded a lot of this work, and understanding that there are very specific genetic alleles that have very high, very different frequencies in different populations. An allele in Duffy is one of the more sort of the poster child of these ancestry informative markers that we can use to actually bin individuals based on true genetic ancestry rather than sort of racial, cultural, or ethnic similarities. But again, undoubtedly, there's an interplay here between race and society and constructs and ancestry and how they impact phenotypes and diseases. And several questions can be asked. Can genetic ancestry be associated with biology and phenotype? And the answer is undoubtedly an astounding yes. I think we can think of a condition or disease such as sickle cell anemia, and I take a moment to make the fact that we have to be careful also about characterizing phenotypes. Everything's not a disease. Some things are just conditions. And it's a condition of an individual in one environment versus another one. Again, sickle cell is a perfect example where individuals who live in a highly malaria-infested area, carrying that allele actually have, it's beneficial. because those individuals can live to ages to reproduce. Individuals who have a normal allele will be affected and can die from malarial infections. You take those individuals out of that environment, put them in an environment where there's no malaria, all of a sudden they have a disease. So we have to be careful about that. And salt retention and hypertension is another classic example. And then we have like diseases and truly deleterious phenotypes like cancer that can be definitely related more to genetic ancestry rather than race. But race, of course, without a doubt, and I know there's social scientists in the audience, I am not the biologist who believes that these differences we see in outcome or health disparities are specifically linked to biology. Some of these things are associated with socioeconomic factors such as poverty, access to health care, environments, and some behaviors that might be associated with diets or lifestyles. And then this complex interplay between genetic ancestry and race. But I want to walk through a few examples where we can start looking at ancestry or race and how it can influence disease risk, and ancestry or race and how it might influence disease outcomes. And the first example will be work that was done in lymphoma by a group out of the ACS, where they published a paper looking at disparities in the adoption of the use of rituximab in patients with diffuse large B-cell lymphoma, where in the early 2000s it was shown that adding this anti-CD20 antibody rituximab to CHOP therapy significantly improved both complete responses and overall response in patients with lymphoma. But they then went on to show that there was a disparity in the adoption and use, showing that individuals who were African American were much less likely to get rituximab added to their chemotherapeutic regimen, and then they were able to go on and show that individuals who had insurance versus those who were uninsured were less likely or more likely to get access to this blockbuster drug. So in this case, one can actually almost say that there's really no genetic ancestry associated with this disparity in outcome, these disparities in outcome or access to these drugs. It's purely access to care. And another example, which I get really excited about, is work that was done in pediatric leukemia. June Yang and Mary Relling, the group out of St. Jude's, and this is, to me, one of the real strong studies that really sort of made a true link between ancestry and biology in a disease. And I think it should be always known as one of the first seminal examples of this. So we know that ALL, 5-year survival rates are really high. Most of these kids do pretty well, but not all of them do, and there are definitely some ethnic or racial differences in survival and outcome, with poor overall survival seen among African American kids and kids of, at the time, were the Hispanic ethnicity. We can use various phrases, Latino, compared to European Americans or Asians. And so June performed a genome-wide association study on about 2,500 kids who had been diagnosed with ALL, and it's important to know that the treatment regimen tends to include initial induction therapy, which can be quite intense, and then consolidation or intensification to really try to drive the cancer into remission. And then after remission, there's then given maintenance. And during maintenance, in some cases, what they'll do is they'll do a real intense infusion for a month or two, and this is called delayed intensification that occurs at the start of maintenance therapy. And this will be important as I talk about summarizing the rest of the study. So he performed principal components analysis from AFI 500K data on these kids, and if you look at the kids, the sort of genetic contribution, the individuals with the red, this is European genetic contribution. Africans are in gray, the African chromosomal material or genetic ancestry. Green is Asian, and then the blue is Native American. And this is where the analysis got really, really interesting. He was actually able to show that kids that had greater than 10% Native American ancestry based on their PCA had a much higher probability of relapse after therapy. And this is relapse after maintenance. And so you can look here and see even kids who self-reported as white but had greater than 10% Native American ancestry also had a much greater probability of relapse. And then when you looked at outcomes based on therapy, when you looked at kids who did not get delayed intensification during maintenance, there was a significant increase in probability, especially when you looked at kids with greater than 10% Native American ancestry. But when you looked at the kids who all got a delayed intensification, you can almost completely eliminate the disparity. So if you have a kid who comes in greater than 10% Native American ancestry, then perhaps that child should always get delayed intensification as part of their maintenance therapy. So again, there's this real cool association between genetic ancestry and a clinical outcome, but through more of a socioeconomic approach and making sure that they get the appropriate therapy, you can completely eliminate that disparity. So I think this is a great example of how we can think about ancestry and population heterogeneity and its influence on disease risk and outcome. And then I want to switch gears and talk a little bit about the work that we've done in multiple myeloma in my lab and in collaboration with scientists from various institutions. I'll walk through that. So myeloma being a disease of plasma cells with a very well-known molecular pathogenesis, and from normal B cell to monoclonal gammopathy of undetermined significance or pre-malignant B cell disease through full-blown myeloma, with a primary genetic lesion being defined by translocations at the IgH locus on chromosome 14 to a series of oncogenes, and downstream progression events including deletion of chromosome 13 or chromosome 13 monosomy, somatic mutations in RAS genes and FGFR, and other secondary events such as amplification of c-Myc. It tends to be a disease of aging, meaning that the median age of diagnosis is around 70, about 30,000 newly diagnosed cases, about 10, 11,000 deaths per year. We've seen this incredible development of blockbuster drugs, particularly the immunomodulatory inhibitors or IMiDs, such as lenalidomide, and proteasome inhibitors such as bortezomib and more recently carfilzomib. With a 5-year overall survival rate before the use of these drugs was only about 37%, and it's increased to almost 50% because of the development of these incredible drugs. But also of importance is that multiple myeloma actually happens to represent one of the most significant cancer health disparities. Looking at incidence rates in males, it actually has the second highest race ratio between African Americans and Europeans, both in males and females. And in looking at death rates, the rate ratio is also among the top killers for both African American men and women. So it's been a very a disease of quite a bit of interest for my research program, where we've got some hypotheses that we're trying to test. One being, although mortality disparities have decreased on the outcome side, there's still a consistent disparity in incidence rate. And that, along with this historical difference in mortality, could suggest a possible genetic role or biological role for these disparities. And we've set out to determine if somatic events in tumors that have been associated with poor outcome in myeloma through other studies might be enriched in tumors from African American patients. We had a large series of about 250 tumors that we profiled in collaboration with the Broad Institute, who performed genome sequencing. My lab did, and in collaboration with Jeff, we did a bunch of work in developing gene expression information and copy number data from these tumors using array technologies. All the data is available publicly through a portal. Interestingly, within our cohort, there are about 16 African American patients, about 180 European American patients. And what we wanted to do was to look at these regions of the genome that had been previously associated with poor outcome or high-risk disease, to see if there were any differences in the frequency of these events and tumors derived from African American and European American papers. And Angie Baker, who's a staff scientist in my lab, was the lead author on this paper, which was a seminal study that was the first to report biological and genomic analysis of tumors from African American patients in comparing to tumors from European American patients. And just looking at the results of the copy number, I won't go through everything in detail, but one thing that jumped out at us was 1Q gain, which is highly associated with a high-risk disease, actually happened to be more frequent in tumors from European American patients than African American patients. It reached statistical significance, and after correction, it went away, but it was still pretty close. And that was an indication that, are we thinking about it the wrong way? Could African American patients actually have tumors that are more associated with favorable outcome? And if they got the right drugs, maybe they'd actually end up doing better. And looking at some other, like the Arkansas high-risk gene expression profile, we didn't see much of a difference there. We looked at 14Q breakpoints. We also saw here that tumors from European American patients were more likely to have breakpoints at 14Q than African American patients, again suggesting that perhaps African American patients have tumors that are more associated with favorable outcome. And in talking to lots of hematologists, oncologists who treat myeloma patients, they all tend to say that African American patients do better when they get the IMiDs and the proteasome inhibitors. So in this case, we believe that African Americans may have tumors that are more associated with favorable outcome, and if they get the appropriate drugs, then perhaps they might actually do as well or better. But there's still this historical difference in incidence, which could suggest that there might be an inherited factor involved in a more frequent development of multiple myeloma in these patients. We've now since begun to apply deeper whole genome sequencing technologies to analyzing these tumors, and of course, they're awesome because we can do very comprehensive genomic interrogation, point mutations across the exome or genome, we can deduce copy number changes, identify gross rearrangements, and if we have RNA, of course, we can do all sorts of cool transcriptional analyses. And just a few slides about the work we're doing, another study through the Multiple Myeloma Research Foundation, which I think it could be a model for cancer genome science going forward. This is a longitudinal study. It's not performing genomic analysis and taking a snapshot of a single sample from a single tumor. In this study, we're actually recruiting patients at diagnosis pre-treatment, and we've recruited 1,000 patients. And we're profiling all of these tumors, normal tumor pairs. Tumors have been enriched for whole genome, whole exome, and RNA sequencing. All of the patients will go on one of 3 treatment regimens, and then at relapse, we're collecting tumors and profiling there as well, so we can really get a sense of the natural history of these tumors during the course of therapy. And this study is really being driven by a really talented scientist at TGen named Jonathan Keats, who I share the PI ship with, and Winnie Liang, who runs the Collaborative Sequencing Center at TGen. And comparing this study to all of the other TCGA and large cancer genome studies, we've generated more data than all except the next-gen sequencing data for all except the breast cancer studies. And just looking at the distribution of RNA, exome, and whole genome, again, this being COMPASS and this being breast. So we've generated quite a bit of data for this project already, and these data will be made publicly available. One of the postdocs in my lab, Zarko Manojlovic, has taken this data and run MUTSIG analysis to look at the mutational status of myeloma tumors and looking at the landscape. And again, we've published some papers previously on the somatic landscape of myeloma in a smaller data set, but overall the mutation frequencies are pretty similar, and we know what the most commonly mutated genes are. But what this study empowered us to do is to be able to look at over 100 African American tumors, making it the largest study to date, and over 500, 600 or so tumors from European American patients. And what we've now done, instead of just using self-identified race, we've begun to extract genetic ancestry information from this. And so this was a principal components analysis done by Zarko, where we took first anchored our PCA using the 1,000 genomes populations, focusing on the Yoruban of Ibadan, Nigeria, the African American population from Southwest, the CEF, or the CEU from Utah, the Mexican from LA, and the Hong Chinese. And so here are the different groups. Here's the Yoruban and the Brown, the Sef, the Mexican from Los Angeles and the Hong. And the African Americans here, you can see this sort of distribution or continuum through sort of we know the admixture between African and European chromosomes. And then we can overlay our myeloma patients here in blue, where we have our European self-identified patients and those that self-identify as African American, and then patients that kind of fall in between different areas of the principal components. And then begin to look at mutation differences based on the PCA, so African Americans that have higher degrees of African ancestry versus those and more centrally distributed in the continuum. And then look at the differences. And so this is just looking at, you know, the genetic alterations across a series of commonly mutated genes, and some interesting things have popped out. For instance, looking at, you know, P53 mutations are much more common in tumors from European American patients than from African American patients. And TP53 loss and mutations are significantly associated with poor outcome in myeloma. So again, these data suggesting that African Americans might have tumors that are associated with more favorable outcome, and then all these mutations that are more common in tumors derived from individuals with African ancestry versus European ancestry. And some of the genes of interest we're looking at is PATCH-D3, not well characterized gene, but with a very significant difference. And I think this group is really interesting because even though we have a lower number of tumors from individuals with African ancestry, we still see these significant differences. We've got almost over 600 tumors here, and so these frequencies should be pretty stable over time. So again, for the first time, really understanding somatic differences in tumors from individuals based on ancestry. And again, you know, looking at the Zarco, looking at the pathways that might be more differentially regulated in individuals, in tumors from individuals from these different ancestral populations. So again, being among the first to really look at this in depth at the tumor biology and relationship to genetic ancestry. And our data, again, would suggest that these African American patients may have tumors with features associated with favorable outcomes. So if they get the right therapies, they actually may have better outcomes. But again, we have not addressed the issues related to incidence, and are hoping to do so through genome-wide association studies in tumors from African American patients using our whole exome data from COMPASS or performing high-density SNP array experiments and GWAS analysis to see if we can identify genetic variants that might be associated with this increase in incidence in myeloma. So hopefully, this review period has sort of walked you through, some aspects of how ancestry can either influence disease risk or outcomes, and it could be race or ancestry. Two examples where access is clearly driving the disparities, where there's a diffuse B-cell lymphoma, multiple myeloma, and then one example where I think ancestry and biology actually is driving outcome disparity in ALL, but you can eliminate that disparity by making sure kids get a very specific treatment regimen as part of their clinical management. So now I want to shift gears a little bit and talk about some of the work we've done in precision medicine, precision oncology. Again, there's a lot I can talk about. We've run a number of clinical trials, the melanoma stand-up to cancer study with Jeff, and studying glioblastoma with Prados in the group, Mike Prados in the group at UCSF, or the study in triple-negative breast cancer we've done with Joyce O'Shaughnessy at Baylor. But yet again, I really want to focus on how population and tumor heterogeneity can influence our interpretation of data related to precision medicine. And this slide just showing the fact that targeted therapeutics are here to stay, and the more we understand about the genomic landscape of tumors, we can provide novel targets to drug developers who can then develop drugs that specifically that hit very specific genomic alterations in these tumors. And I think one of the things that really is awesome is the fact that these drugs don't have to be indicated for a single disease type. We know imatinib works both in BCR-ABL positive CML as well as KIT and PDGFR mutated GIST. We've seen activity with EGFR inhibitors, both non-small cell lung cancer and colon cancer. And more recently, we've seen the development of PARP inhibitors and work that we did with Rulshanine and Ken Pienta, showing that BRCA2 homozygous deletions occur at frequencies higher than we would have once thought in castration-resistant prostate cancer. And these tumors respond to PARP inhibitors. And there was a fast-track, expedited approval of olaparib for the treatment of a subset of castration-resistant prostate cancers. And then, I'd be remiss to not say that this new sort of, it's really not new, but a newly adopted approach of immune checkpoint inhibitors treatment modality and targets like PD-1 or PD-L1 on the tumors, PD-1 or CTCLA4 in the T cells, and the incredible outcomes that we're seeing with these immune checkpoint inhibitors. Doesn't work for everybody, but in those tumors where it does work, and we have some biomarkers where it might work, like hypermutation, expression of these biomarkers, or the expression of neoantigens. So this growing laundry list of genomic alterations and targeted therapies has led us to this new revolution of precision medicine, and where we can profile a tumor, identify the appropriate alterations, and then perhaps select a targeted therapy for that patient's cancer. These events, of course, these genes are mutated by various mechanisms. We know the oncogenes in many cases can be amplified or mutated or overexpressed, tumor suppressors deleted, mutated, or hypermethylated. And we also know that some of these genes are altered by breakpoints, translocations, leading to the generation of oncogenic fusions that can also be targeted. So, through collaborations, a lot of folks at TGen, and in partnership primarily with my good friend and colleague, David Craig, God, I can't say enough awesome things about David and the work that he's done to help build this out. But building out this algorithm, and I have to give credit to, the Stand Up to Cancer team that helped us derive this and submit a exemption for a device to the FDA in support of our Jeff's Stand Up to Cancer study. But, starting with patient and doc and consent and biopsy collection of specimens, and sending those specimens through quality assessment of the analytes and sequencing and bioinformatics analysis, and then essentially merging the somatic information to the drug space, and then generating these reports, holding molecular tumor boards to vet that information, and then provide what we feel is the most appropriate therapy for that patient based on the molecular profile. Part of this was building out an incredibly standardized and validated platform, part of which was the creation of a controlled access clinical portal. And again, this is David Craig, my just tremendous friend and colleague, where each patient can, a clinical research nurse can go in and create a new patient, fill out a form with a controlled lexicon and vocabulary, standardized clinical annotations, and then from this be able to auto-generate a report that can be used as part of the molecular tumor boards and the sequencing. I don't get any money from Illumina, but I have their machine here. Just want to make sure that's said. And then we have, again, David's team building out just incredible bioinformatics framework to support everything from data collection, data management, data analysis, secondary analysis, generation and annotation of the information, creating the reports that can then be vetted by the tumor board, and an incredible establishment of a relationship with Dell Computing, a lot of hard work that Jeff did that has supplied us with an incredible high-performance computing environment to support this work. And then, the process of comparing each patient's somatic tumor profile to a relational database developed by Jeff Kiefer and more recently Sarah Byron, and using heuristic programs and algorithms to match the mutation profile with the drug gene relation database, and we can sort of modify or tune the pharmacopoeia however we want based on the study to generate reports that will have the drug, other information like is there an oral formulation for the drug. Sometimes it's really important. The alteration driving that relationship, is it a positive or negative indication, and whether or not there are clinical trials available. We built a CLIA lab, fully validated, so it's CLIA certified, CAP accredited. I have to give credit to Janine Labello and Lisa Bombard-Reardon, who were the lab directors, Mary Ellen Ahern, who was the lab manager, who helped us build this, this clinical laboratory. And Jeff and Pat had to submit this document to the FDA in support of their clinical trial. And I have a picture of their faces before and after this process. And I think there were a number of babies born, some divorces, and a few people quit after this document was submitted. But boy, the overall benefits of having this in place have been tremendous for us, because we feel like we're thought leaders in this space, and being able to validate a platform such that it meets the rigorous and stringent guidelines for CLIA, CAP, and the FDA. A number of papers have been published on methodologies, as well as the results of some of these trials. The triple-negative breast cancer study being the first, and was the most cited paper in Molecular Cancer Therapeutics in 2014. Amazing study led by Sarah Byron with Mike Prados and glioblastoma. Even on the pediatric side, work with Lenny Cinder at Children's Hospital Orange County, led by my postdoc Troy McEachran, was recently promoted to assistant professor. And I'm going to talk a lot about the studies we've done in cholangiocarcinoma, and of course the work that Jeff and Pat are leading in looking at BRAF V6 BRAF V600E mutated melanomas, and even some papers related to methods and for validation. We just had this in Nature Reviews, and just sort of reviewing the translation of RNA sequencing into the clinic. We have this paper where we have a set of synthetic oligos that mimic oncogenic fusions that can be used, spiked into a sample in a clinical lab, and used to validate the detection of oncogenic fusions from RNA-seq data. And then just a week or two ago, this came out in collaboration with Illumina, as well as Mark O'Mara's group in Vancouver, where we have a normal tumor cell line pair, COLO 829, where we've all sequenced this in a clinical laboratory setting, and actually developed a set of truth variants that can be used or compared to in any clinical laboratory to put this cell line pair forward as a somatic reference standard for clinical sequencing. And so now moving on to population heterogeneity, I think one of the issues we face in precision medicine today is the fact that, in many cases we want to just be able to use a tumor sample, because in using this method, we really don't interfere with the clinical standards and approaches in pathology labs, right? We're not asking them to give us a piece of fresh tissue and sort of alter their day-to-day workflows. But some feel that requesting a sample, and some feel that requesting a sample for constitutional DNA is not always feasible, and complicates or slows the testing. But there may be high risk of false positives, and this is kind of where I want to focus this talk. And this is very important, I think, from the population standpoint, as some populations have higher genetic variation, so much more likely to have private variants in their genome, and those private variants can sometimes trigger drug rules. And we've seen it many times, and just some examples, and I'm not going to name labs, I'm just going to say Lab E, Lab A, and Lab B. This lab doing somatic analysis on the same tumor or the same DNA that was used for tumor-only analysis, where this lab had normal tumor, knew or was able to state specifically that was a germline BRCA mutation. And looking at the variants of undetermined significance on this list, almost 10 to 12 of these variants actually were germline variants. They had nothing to do with, these were not tumor-acquired somatic mutations. Another case, colorectal cancer, same situation. Another patient with metastatic triple-negative breast cancer, where our lab detected a frame shift mutation in PIK3R1, which is therapeutically actionable, happened to be missed by the other lab. We won't go into that very much. But there were two other mutations on the front page of that report, one of which triggered a drug rule. And interestingly, we were able to show, because we did normal tumor pair, that actually was those two were germline variants. One of these genes, Notch, even though it didn't trigger drug rule here, later could have, which would have been the use of gamma secretase inhibitors, for which one of the side effects is incredible diarrhea. And so we want to make sure that we're doing this appropriately and that individuals, again, from populations that have higher degrees of genetic variation, where data may not necessarily be in the public databases for filtering, can have more of these false positives on their reports. And so it's work done by Rebecca Halpern in David's lab and Zarko in my lab. And we took a series of patients, and we had African Americans, European Americans, and performed principal components analysis and looked at the number of mutations that would have been sort of from a tumor-only analysis if you just use filtering using publicly available SNP data, showing that you would have had a much higher number of false positives and individuals who have higher degrees of African ancestry versus individuals of European ancestry. And looking at the number of mutations, again, the average being 242 here versus 125, and then looking at European versus African American. This table's not right, but essentially what we're seeing here is that if you were African American, there'd be an average of 240 variants on average, versus 125 if you were European American, and 2.5 or 3 would have triggered a drug rule in an African American versus 1.5 of those false positive drug rule triggering variants would have been on your report if in the case of European Americans. So again, and this is a statistically significant difference. So there's definitely an ancestral bias to this, and we've been playing around with optimizing approaches using probabilistic approaches. And essentially what we've done is we've sequenced a series of tumors really deep, done some dilution series, and came to the conclusion that if you have more normal contaminated stroma in the specimen, you can use the germline variants because whether it's tumor or germline variants should have an allele frequency of about 50-50. In a pileup if it's a heterozygous variant. But if it's a tumor variant and there's normal contaminated stroma, those heterozygous somatic variants will have an allele shift. And you can use that information to reduce the number of false positives. And if you sequence higher, meaning if you have a really high quality sample that's like 90% tumor, you have to sequence upwards of 1,600 to 3,200x to be able to identify these allele shifts. And so we've played, David and Rebecca and the team have essentially came up with some algorithms where if this was a triple negative breast cancer from a Ghana African patient, you have almost 350 false positives if you use filtering only. But if you use our approach, you can significantly decrease the number of false positives that would end up on an approach. And here Ghana and European American triple negatives and African American triple negatives and a patient with glioblastoma that had Latino ancestry. So I'm going to use the final few minutes of my presentation to talk about tumor heterogeneity and its impact on precision medicine. I think many of us in cancer have followed the work of Charlie Swanton and the group over at Cambridge and in the UK, the Sanger Center, where they took a sample from a kidney cancer patient and chopped it up into various pieces and then looked at the regional profile of each of these specimens. And they also had a chest wall metastasis that they looked at as well. They were able to show that one of those specific regions, region R4, which was this region, had mutations that were also available in the METs, but none of the other lesions from that tumor did, suggesting that this tumor sort of sat in the middle between the primary and the METs. And so they were able to draw these phylogenetic trees. Just amazing work that showed at the gene expression level, R4 also mimicked the METs more than any of the other from the primary renal lesion. And so we've worked out some algorithms. So this is one of our COMPASS multiple myeloma patients. And Jonathan Keats has built out bioinformatics tools that will allow us to look at clonal variants in the tumor. So in this particular patient, we picked up 3 different clones. This is looking at the sort of the bimodal or trimodal distribution of heterozygous variants, which indicate various subclones. And so in this particular patient, looking at time point 1 and time point 2, the distribution of these clones is pretty similar, meaning this clone remained the dominant clone throughout the course of this patient's clinical management. Yet another patient, you see very significant clonal dynamics occurring, where at diagnosis you have this red and blue, these red and blue clones that are the primary, and this low-lying green clone was also low at this time point, but became among the dominant clones, and the blue clone goes away over various courses of treatment. So this tumor heterogeneity and this shifting and flowing of various clones under various therapeutic conditions is going to provide us with information on understanding what mechanisms are associated with response or resistance to the various therapies. And then finally, work we've done in cholangiocarcinoma or bile duct cancer, tumors of the bile duct. They can occur sporadically or be associated with liver fluke infection. We're primarily focusing on the sporadics, and it can be very difficult to treat. And we published this paper with Alan Bryce and Mitesh Borat at the Mayo Clinic in Scottsdale, where we showed the integrated genomic characterization and clinical sequencing identified therapeutically actionable context around FGFR and EGFR pathways and treatment. One patient with an ERFI1 homozygous deletion. ERFI1 is a very obscure gene, was not on many of the cancer panels that people were using in clinical laboratories. But it's critically important because its role is to prevent dimerization of ERB receptors. So I mean, it can be has to be one of the most important tumor suppressors. But understanding this patient had loss, hypothesizing that it was leading to hyperactivation of the ERB signaling, EGFR signaling, tumor before erlotinib, tumor after erlotinib. This is looking at metabolic activity, just a dramatic response because we selected erlotinib because of the hypothesis of EGFR activation. We also identified tumors that had FGFR2, oncogenic fusions and translocations, treating those patients with FGFR inhibitors, looking at densitometric scans before ponatinib, which is a select FGFR inhibitor, and tumor after, tumors just melting away in these patients. So moving forward and thinking more about the concept of heterogeneity in a different context, patient comes in with advanced cholangiocarcinoma, sample sent for clinical sequencing. Looking at the copy number data, we saw this event on chromosome 8, which was characterized as leading to an oncogenic fusion that encompassed NRG1. NRG1 happens to be the ligand for ERB receptors leading to increased phosphorylation activation. Again, more evidence that in this tumor type, there's a context around activated ERB receptor signaling, and ERB fusions have been seen in other tumors, not specifically this one, but what they showed was that these fusions leaded to activation of HER2 and HER3. We also identified yet another ERFI1 inactivating event in this tumor. So we had activation of the ERB receptor pathway on both sides, both activation, hyperactivation through NRG and loss of MYC6 ERFI1. The discussion of the tumor board focusing around ERB receptor signaling, not wanting to target any of the specific receptors, but thinking about targeting multiple receptors by using a drug that would prevent dimerization, that drug being pertuzumab. This is liver cancer before, liver cancer after. Just an incredible response to this, but it was a transient response. There was a progression after several months on therapy. We were actually able to collect a biopsy at progression, and looking at the copy number, the 8 event went away, and this blazing amplification of EGFR pops up that wasn't there in the initial tumor, right? So treat with pertuzumab, we get rid of the NRG1 clone. This new clone has arisen that has this incredible amplification of EGFR. Significant response to erlotinib, but again, it was transient, and now we get a third biopsy, right? So what happens next, right? You've been on pertuzumab, had a response. You then go on erlotinib, have a response. Cancer comes back. So what happens? Brrr, right? Is this going to change? I think I ran out of. Will this work? Acknowledgements. What did I do? You guys get to see my talk backwards to forwards. Okay, here we go. One more. Can you do one more? No, I don't. One more. Go forward. It shouldn't. I am. So I've like pressed it like 80 times in both directions. Don't press the button. Guess what happens? The first clone comes back. So you've got this clonal tides. So pertuzumab kills this clone, erlotinib kills this clone, but the first clone came back. But what happens in clinical oncology at progression? You never give a previous treatment because in the oncologist's mind, the patient has failed that treatment, right? And so we have to take these aspects of tumor heterogeneity in, I can't get this move, into context of precision medicine. And I think it's complicating our ability to see improved outcomes using, this incredible clinical approach. So in summary, population and tumor heterogeneity in genome science and precision oncology exists. There is race and ancestry, and they can be related but are not necessarily the same. I think I've shown that. There may be an enrichment of certain biological factors that can be associated with biology and phenotypes and outcomes. Research into uncovering these factors will help us better understand disease etiology within various populations and could provide information on how to best approach clinical management. Development of tools and methodologies can help to improve the performance of precision medicine, which can have critical impact on clinical interpretation results. And I think I've shown that particularly in ancestral populations that have not undergone significant bottlenecks where there are lots of genetic variants. And tumor heterogeneity, of course, exists and is a confounder in current precision medicine approaches, and better tools and methods will allow us to better sample the entirety of a patient's cancer instead of taking snapshots of individual biologies to uncover these clonal aspects of these tumors that are primed to induce relapse. So, in conclusion, one more. Gonna go? Should I touch the button? Who cares? Acknowledgements. Boy, this thing was full of people's names. It was all me. But, specifically, of course, David Craig and all the work that he's done. He's been an incredible collaborator and partner in a lot of this work, Jonathan Keats and Winnie Liang, and developing a lot of our sequencing infrastructure there at TGen. Can't say enough about the support that Jeff has given me through the career to have the freedom to approach these high-risk studies that have, I believe, allowed me to apply knowledge and science and genomics to improve clinical care for patients. And then our clinical partners, Mitesh and Alan at Mayo Clinic, Pat LaRusso at Yale, Dan Von Hoff and the group at Scottsdale Health, who Dan has been sort of the godfather of precision medicine at TGen, and then all of the funding agencies, MMRF, the Prostate Cancer Foundation, Komen for the Cure, the Ben and Catherine Ivy Foundation, the NIH, and the NCI. So I'll stop there. Thank you. I don't know if there's
a couple of quick questions.
If there are any, I'd love to engage the audience.
Go ahead, Thomas.
So,
Thomas.
this neuroregulatory NRG1, this gene actually localizes at the site of a common copy number variant. And we and others have shown that these are sites where the chromosome breaks, and this is correlated to either translocations or sites of copy number changes if you do CGH.
Absolutely, no, absolutely. There's a really interesting observation that we've made in triple-negative breast cancer. I don't want to give it away yet because I think it's going to be seminal. But we've seen breakpoints in a very specific region of the genome encompassing a very important tumor suppressor, only in tumors from African American women so far. And my hypothesis is that there's some genetic haplotype or some that's more frequent in individuals of African ancestry, that's leading to these breakpoints occurring at that very specific region of the genome. So, as part of our ongoing studies, our hope is to better understand that, to be able to, utilize publicly available data like the data you guys are generating to look at where these breakpoints are occurring, and to understand more about the genetic contribution and haplotypes and how they relate to specific alterations occurring in specific somatic events occurring at specific regions of the genome. I think that it's been a lost opportunity in the cancer genome space, that there really hasn't been enough integration between the germline community and the somatic community to really understand how the germline influences somatic alterations in cancer. And I think that could be one of the more exciting opportunities for us going forward in understanding cancer initiation and progression and outcomes in different groups of people.
Maybe I can ask you a question. So
Sure.
for the multiple myeloma story, you mentioned the African Americans, they actually have a good, favorable prognosis signature, but their prognosis, is actually worse. So have you looked at the treatment, they have received?
Well,
that's the correlation.
That's the amazing thing about the COMPASS study, is that it's longitudinal, and we know treatment up front, and we know treatment along the patient's clinical management. So we don't yet have that information, but we will by the end of the study, because we'll have arguably the only data set that will allow us to do that, that's powered enough to make those correlations, to be able to say, if you have these events and you have favorable outcome, but you didn't get these 3 specific regimens, you're likely to do worse and you're likely to do better. So we'll be able to make those correlations because of the design of that study.
If no further questions, I'd just like to thank you very much for this wonderful lecture.
Thank you.
There will be a reception right now in the NH library, and as a token of appreciation, we're going to give this to John. It's a aerial photo of NH, and you're welcome to sign this around the frames. This will be in the library. At
Where's
the
Building 9? That's
Where's
what I'm
Building
looking
9? It's
for.
somewhere right
There's
here.
history in the basement of Building 9.
It
I need to go take a picture of it before they tear it down. Thanks, Paul.
Be there. Yeah.
Thanks, everybody. All right.